Hello everyone, my name is Lotfi, and in this video we will be talking about buffers, and these are the main subjects for this video. And so without any further ado, let's get started with our video. As you might know, the internal pH of most living cells is close to neutral, or the pH of 7. And even a slight change in the pH can be harmful because the chemical processes of the cell are very sensitive to the concentration of hydrogen and hydroxide ions. Okay, and as we will learn in the future, proteins that do most of the cell's work are very sensitive to the shifting of pH of their solution. Okay, so when their pH of their environment or solution changes, their 3D structure, which is very important for their function, changes as well, and thus they can't do their work properly. Okay, for example, the pH of human blood is very close to 7.4, and this means that our blood is slightly basic, and so the proteins and cells that are present in our blood function best at the pH of 7.4, and thus a person cannot survive more than a few minutes if the blood pH drops to 7 or rises to 7.8. Okay, and so our blood should have a chemical system that maintains a stable pH. Okay, and you might be asking yourself, why does it matter if our blood pH shifts just 0.4 degrees? And if I were to answer your question, I would say, take for example, if we add 0.01 moles of a strong acid to a liter of pure water, the pH would drop from neutral, or pH of 7, to the pH of 2. Okay, so it becomes more acidic. So it is very important for us to not let that happen in our own body. Okay, and our body has invented a way to not let that happen. For example, if we add the same amount of acid that we did in the water example that we discussed a few seconds ago, I think, our blood pH would decrease from 7.4 to 7.3, and don't experiment this at home or anywhere, because I just told you the answer, and now I'm going to tell you how this happens. So this phenomena happens because of the presence of substances that are called buffers, okay, which they allow biologists to maintain a relative pH in fluids despite the addition of acids and or bases. Okay, and the better definition of buffers are substances that minimize the changes in the concentration of H plus or hydrogen and hydroxide ions in a solution, and you can see the both definitions in this page. And so buffers do this by accepting hydrogen ions from the solution when they are in excess and donate the ions when they have been depleted from the solution. And also most buffer solutions contain a weak acid and its corresponding base, which combine reversibly with hydrogen ions. Okay, and this is, in this picture you can see a buffer, okay, which gains a hydrogen ion and donates a hydrogen ion. Okay, and as shown by the two arrows, this reaction is a reversible reaction. And have in mind that several buffers contribute to pH stability in human blood, and many other biological solutions, other solutions that are used by other animals and other biological or living things. Okay, so one example would be carbonic acid or H2CO3, okay, which forms when CO2 reacts with water in the blood plasma. Okay, and so carbonic acid dissociates to form a bicarbonate ion and a hydrogen ion, as you can see in the reaction written above. Okay, and the chemical equilibrium between carbonic acid and bicarbonate acts as a pH regulator. And as I told you in the previous slide, when the solution is depleted of hydrogen ions, in this case carbonic acid dissociates and forms bicarbonate and a hydrogen ion, okay, and vice versa. In conclusion, the carbonic acid and bicarbonate system consists of an acid and a base in equilibrium with each other, and like this buffer, other buffers also are also acid-base pairs. Okay, and so that was all for this video. I hope you enjoyed it, and if you did, please hit the like button, and as always, until the next video, happy learnings to you all.